Hello students, welcome back to Cyber School. This is the second part of the photosynthesis process video series. If you haven't watched the first part of the series, you can't get an idea about this video. I have included that video link in this video description. Go back and watch that first, and subscribe the channel. Today I talk about development of the idea, the overall reaction of photosynthesis. Let's begin. Development of the idea. The study of photosynthesis began in 1771 with observations made by the English clergyman and scientist Joseph Priestley. Priestley had burned a candle in a closed container until the air within the container could no longer support combustion. He then placed a sprig of mint plant in the container and discovered that after several days the mint had produced some substance, later recognized as oxygen, that enabled the confined air to again support combustion. In 1779, the Dutch physician Jan Ingenhousz expanded upon Priestley's work, showing that the plant had to be exposed to light if the combustible substance, oxygen, was to be restored. He also demonstrated that this process required the presence of the green tissues of the plant. In 1782, it was demonstrated that the combustion-supporting gas, oxygen, was formed at the expense of another gas, or fixed air, which had been identified the year before as carbon dioxide. Gas exchange experiments in 1804 showed that the gain in weight of a plant grown in a carefully weighed pot resulted from the uptake of carbon, which came entirely from absorbed carbon dioxide, and water taken up by plant roots. The balance is oxygen, released back to the atmosphere. Almost half a century past the concept of chemical energy had developed sufficiently to discovery, in 1845, that light energy from the sun is stored as chemical energy in products formed during photosynthesis. The overall reaction of photosynthesis. In chemical terms, photosynthesis is a light-energized oxidation-reduction process. Oxidation refers to the removal of electrons from a molecule, reduction refers to the gain of electrons by a molecule. In plant photosynthesis, the energy of light is used to drive the oxidation of water, H2O, producing oxygen gas, O2, hydrogen ions, H+, and electrons. Most of the removed electrons and hydrogen ions ultimately are transferred to carbon dioxide, CO2, which is reduced to organic products. Other electrons and hydrogen ions are used to reduce nitrate and sulfate to amino and sulfhydryl groups in amino acids, which are the building blocks of proteins. In most green cells, carbohydrates, especially starch and sugar sucrose, are the major direct organic products of photosynthesis. The overall reaction in which carbohydrates, represented by the general formula, CH2O, are formed during plant photosynthesis can be indicated by the equation. This equation is merely a summary statement, for the process of photosynthesis actually involves numerous reactions catalyzed by enzymes, organic catalysts. These reactions occur in two stages: the light stage, consisting of photochemical, i.e., light capturing, reactions, and the dark stage, comprising chemical reactions controlled by enzymes. During the first stage, the energy of light is absorbed and used to drive a series of electron transfers, resulting in the synthesis of ATP and the electron donor-reduced nicotine adenine dinucleotide phosphate, NADPH. During the dark stage, the ATP and NADPH formed in the light-capturing reactions are used to reduce carbon dioxide to organic carbon compounds. This assimilation of inorganic carbon into organic compounds is called carbon fixation. During the 20th century, comparisons between photosynthetic processes in green plants and in certain photosynthetic sulfur bacteria provided important information about the photosynthetic mechanism. Sulfur bacteria use hydrogen sulfide, H2S, as a source of hydrogen atoms and produce sulfur instead of oxygen during photosynthesis. The overall reaction is: in the 1930s, Dutch biologist Cornelis van Niel recognized that the utilization of carbon dioxide to form organic compounds was similar in the two types of photosynthetic organisms. Suggesting that differences existed in the light-dependent stage and in the nature of the compounds used as a source of hydrogen atoms, he proposed that hydrogen was transferred from hydrogen sulfide, in bacteria, or water, in green plants, to an unknown acceptor, called A, which was reduced to H2A. During the dark reactions, which are similar in both bacteria and green plants, the reduced acceptor, H2A, reacted with carbon dioxide, CO2, to form carbohydrate, CH2O, and to oxidize the unknown acceptor to A. This putative reaction can be represented as. Van Niel's proposal was important because the popular, but incorrect, theory had been that oxygen was removed from carbon dioxide, rather than hydrogen from water, releasing oxygen, and that carbon then combined with water to form carbohydrates, rather than the hydrogen from water combining with CO2 to form CH2O. By 1940, chemists were using heavy isotopes to follow the reactions of photosynthesis. Water marked with an isotope of oxygen, 18O, was used in early experiments. Plants that photosynthesized in the presence of water containing H218O produced oxygen gas containing 18O. Those that photosynthesized in the presence of normal water produced normal oxygen gas. These results provided definitive support for Van Niel's theory that the oxygen gas produced during photosynthesis is derived from water. Okay, students, this is the end of today's video. Thanks for watching. Join the next part of this video. If you learned something from this, please feel free to comment below. If this is good content, hit the like button and share it with your friends. Goodbye. Have a nice day.